A very good day to all the viewers. The topic of the discussion here is chemistry of life. Introduction. Life originated on Earth million years ago, and since then innumerable varieties of living beings are evolved. To understand the mechanism of life, or how living organisms manage to reproduce, grow, move, think, eat and do whatever it is that they are doing, biologists can apply chemistry and physics to the study of life. The important foundation for any biologist who wants to understand mechanisms are based on the answers to the following questions. What is an element? What is a molecule? What is a macromolecule? And why is carbon important? Chemistry of life. Life is a chemical process. All aspects of living creatures have a chemical basis. An understanding of life requires an understanding of the chemical basis of life. All things are made of matter. Matter, in turn, is made up of chemical substances. In this lesson, we will learn about the chemical substances that make up living things. A chemical substance is a matter that has a definite composition. It also has the same composition throughout. A chemical substance may be either an element or a compound. Elements. An element is a pure substance. It cannot be broken down into other types of substances. Each element is made up of just one type of atom. An atom is the smallest particle of an element that still has the properties of that element. There are almost 120 known elements. The element most important to life is carbon, abbreviated as capital C. Compounds. A compound is a substance that consists of two or more elements. A compound has a unique composition that is always the same. The smallest particle of a compound is called a molecule. Consider water as an example. A chemical reaction is needed to form a compound. Another chemical reaction is needed to separate the substances in a compound. A compound found mainly in living things is known as an organic compound. Organic compounds make up the cells and other structures of organisms and carry out life processes. Carbon is the main element in organic compounds, so carbon is essential to life on Earth. Without carbon, life as we know could not exist. The significance of carbon. Why is carbon so basic to life? The reason is carbon's ability to form stable bonds with many elements, including itself. This property allows carbon to form a huge variety of very large and complex molecules. In fact, there are nearly 10 million carbon-based compounds in living things. However, the millions of organic compounds can be grouped into just 4 major types, that are carbohydrates, lipids, proteins and nucleic acids. Carbohydrates. Carbohydrates are the most common type of organic compound. A carbohydrate is an organic compound such as sugar or starch and is used to store energy. Like most organic compounds, carbohydrates are built up of small repeating units that form bonds with each other to make a larger molecule. In the case of carbohydrates, the small repeating units are called monosaccharides. Monosaccharides. Monosaccharides are the most basic units of carbohydrates. They are the simplest form of sugar and are usually colorless, water-soluble, crystalline solids. Some monosaccharides have a sweet taste. Examples of monosaccharides include glucose, that is dextrose, fructose, that is levulose, and galactose. Fructose is found in fruits, whereas glucose generally results from the digestion of other carbohydrates. Glucose is used for energy by the cells of most organisms. Monosaccharides are the building blocks of disaccharides and polysaccharides. With few exceptions, example deoxyribose, monosaccharides have the chemical formula CXH2OY, where conventionally X is greater than or equal to 3. Monosaccharides can be classified by the number X of carbon atoms they contain into diose, triose, tetrose, pentose, hexose, heptose, and so on. The most important monosaccharide, glucose, is a hexose. Examples of heptoses include the ketoses, menoheptulose, and pseudoheptulose. Monosaccharides with 8 or more carbons are rarely observed as they are quite unstable. Disaccharides. A disaccharide or biose is the carbohydrate which is formed when two monosaccharides undergo a condensation reaction which involves the elimination of a small molecule such as water from the functional groups only. Disaccharide is one of the 4 chemical groupings of carbohydrates that are monosaccharide, disaccharide, oligosaccharide, and polysaccharide. The most common types of disaccharides are sucrose, lactose, and maltose. They have 12 carbon atoms with the general formula C12H22O11. The differences in the disaccharides are due to atomic arrangements within the molecule. Polysaccharides. Polysaccharides are polymeric carbohydrate molecules composed of long chains of monosaccharide units bound together by glycosidic linkages and on hydrolysis give the constituent monosaccharides or oligosaccharides. They range in structure from linear to highly branched. Examples include storage polysaccharides such as starch and glycogen and structural polysaccharides such as cellulose and chitin. Polysaccharides are often quite heterogeneous, containing slight modifications of the repeating unit. Depending on the structure, these macromolecules can have distinct properties from their monosaccharide building blocks. They may be amorphous or even insoluble in water. When all the monosaccharides in a polysaccharide are the same type, the polysaccharide is called a homopolysaccharide or homoglycan, but when more than one type of monosaccharide is present, they are called heteropolysaccharides or heteroglycans. Polysaccharides are an important class of biological polymers. Their function in living organisms is usually either structure or storage related. Starch, that is a polymer of glucose, is used as a storage polysaccharide. In animals, the structurally similar glucose polymer is the more densely branched glycogen, sometimes called animal starch. Glycogen's properties allow it to be metabolized more quickly, which suits the active lives of moving animals. Cellulose and chitin are examples of structural polysaccharides. Cellulose is used in the cell walls of plants and other organisms and is said to be the most abundant organic molecule on earth. Chitin has a similar structure but has nitrogen containing side branches, increasing its strength. It is found in arthropod exoskeletons and in the cell walls of some fungi. Lipids. A lipid is an organic compound such as fat or oil. Organisms use lipids to store energy, but lipids have other important roles as well. Lipids consist of repeating units called fatty acids. There are 2 types of fatty acids. They are saturated fatty acids and unsaturated fatty acids. Saturated fatty acids. In saturated fatty acids, carbon atoms are bonded to as many hydrogen atoms as possible. This causes the molecules to form straight chains as shown in this figure. The straight chains can be packed together very tightly, allowing them to store energy in a compact form. This explains why saturated fatty acids are solids at room temperature. Animals use saturated fatty acids to store energy. Unsaturated fatty acids. In unsaturated fatty acids, some carbon atoms are not bonded to as many hydrogen atoms as possible. Instead, they are bonded to other groups of atoms. Wherever carbon binds with these other groups of atoms, it causes chains to bend, as shown in the figure here. The bent chains cannot be packed together very tightly, so unsaturated fatty acids are liquids at room temperature. Plants use unsaturated fatty acids to store energy. Types of lipids. Lipids may consist of fatty acids alone, or they may contain other molecules as well. For example, some lipids contain alcohol or phosphate groups. They include number 1. Triglycerides. Triglycerides are the main form of stored energy in animals. Number 2. Phospholipids, that are the major components of cell membranes. And number 3. Steroids. Steroids serve as chemical messengers and have other roles. Proteins. A protein is an organic compound made up of small molecules called amino acids. There are 20 different amino acids commonly found in the proteins of living things. Small proteins may contain just a few hundred amino acids, whereas large proteins may contain thousands of amino acids. Protein structure. Most proteins fold into unique three-dimensional structures. The shape into which a protein naturally folds is known as its native conformation. Although many proteins can fold unassisted simply through the chemical properties of their amino acids, others require the aid of molecular chaperones to fold into their native states. Biochemists often refer to 4 distinct aspects of a protein's structure. They are primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure. The primary structure of a protein refers to the linear sequence of amino acids in the polypeptide chain. The primary structure is held together by covalent bonds such as peptide bonds, which are made during the process of protein biosynthesis or translation. A protein is a polyamide. Secondary structure. It is regularly repeating local structures stabilized by hydrogen bonds. The most common examples are the alpha helix and beta sheet. Because secondary structures are local, many regions of different secondary structure can be present in the same protein molecule. Tertiary structure. The tertiary structure is the overall shape of a single protein molecule, the spatial relationship of the secondary structures to one another. It is generally stabilized by non-local interactions, most commonly the formation of a hydrophobic core, but also through salt bridges, hydrogen bonds, disulfide bonds, and even post-translational modifications. The term tertiary structure is often used as synonymous with the term fold. Quaternary structure. This structure is formed by several protein molecules or polypeptide chains, usually called protein subunits, which function as a single protein complex. Proteins are not entirely rigid molecules. In addition to these levels of structure, proteins may shift between several related structures while they perform their functions. Functions of proteins. Like other biological macromolecules such as polysaccharides and nucleic acids, proteins are essential parts of organisms and participate virtually in every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to metabolism. Proteins also have structural or mechanical functions such as actin and myosin in muscle and the proteins in the cytoskeleton, which form a system of scaffolding that maintains the cell shape. Other proteins are important in cell signaling, immune responses, cell adhesion and the cell cycle. Proteins are also necessary in animals' diets since animals cannot synthesize all the amino acids they need and must obtain essential amino acids from food. Through the process of digestion, animals break down ingested protein into free amino acids and are then used in metabolism. For example, human red blood cells contain a protein called hemoglobin, which binds with oxygen. Hemoglobin allows the blood to carry oxygen from the lungs to cells throughout the body. Nucleic acids. A nucleic acid is an organic compound such as DNA or RNA that is built of small units called nucleotides. Many nucleotides bind together to form a chain called a polynucleotide. The nucleic acid DNA, that is deoxyribonucleic acid, consists of two polynucleotide chains. The nucleic acid RNA, that is ribonucleic acid, consists of just one polynucleotide chain. Structure of nucleic acids. Each nucleotide consists of three smaller molecules. They are sugar, phosphate group and nitrogen base. In the figure on the screen, we see that the sugar of one nucleotide binds to the phosphate group of the next nucleotide. These two molecules alternate to form the backbone of the nucleotide chain. The nitrogen bases in a nucleic acid stick out from the backbone. There are four different types of bases. They are cytosine, adenine, guanine and either thymine in DNA or uracil in RNA. In DNA, bonds form between bases on the two nucleotide chains and hold the chains together. Each type of base binds with just one other type of base. Cytosine always binds with guanine and adenine always binds with thymine. These pairs of bases are called complementary base pairs. The binding of complementary bases allows DNA molecules to take their well-known shape called a double helix, which is as displayed here on the screen. A double helix is like a spiral staircase. The double helix shape forms naturally and is very strong, making the two polynucleotide chains difficult to break apart. Roles of nucleic acids. DNA is found in genes and its sequence of bases make up a code. Between start and stop codon, the code carries instructions for the correct sequence of amino acids in a protein. RNA uses the information in DNA to assemble the correct amino acids and help make the protein. The information in DNA is passed from parent cells to daughter cells whenever cells divide. The information in DNA is also passed from parents to offspring when organisms reproduce. This is how inherited characteristics are passed from one generation to the next.
Summary. Now let us summarize this topic. Living things consist of matter which can be an element or a compound. A compound consists of two or more elements and forms a result of a chemical reaction. Biological macromolecules are defining the properties of cells. These molecules include proteins, nucleic acids, carbohydrates and lipids. The properties they convey are enzymatic activity or metabolism, genetic inheritance, reproduction, cell growth, energy storage and conversion and interaction with an environment. All living organisms use the same 4 types of macromolecules for cellular metabolism and reproduction. Together, they illustrate the similarities of life on Earth. The way they are used in different forms and combinations explains today's variety or biodiversity. Both aspects, sameness and variety, are the result of biological evolution. For instance, biological molecules are made of carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus and other ionic species such as sodium, potassium, chloride, calcium, magnesium, iron, copper, cobalt, manganese, selenium, etc. Thank you.